So once light actually makes it through, the cornea at the front here, right, works its way past the iris, through the colored part of the eye, through the pupil, which is, either constricted or dilated appropriately to let in the right amount of light, and then, passes through the lens, which you are, flexibly adjusting, stretching or relaxing to bend the light rays appropriately, to focus the image back here on this, set of cells back here called the retina. Well, then it's going to actually strike what are called photoreceptors, which are located at the very back of the retina. So the most posterior portion of this white circle here. This is actually where the cells are located that are gonna actually carry out what we call stimulus transduction, like conversion of the electromagnetic radiation, the actual absorption of that electromagnetic radiation, and conversion into some kind of neuronal response, to start sending that information sort of back to the rest of the brain about what it is that you are detecting with your eyes, right? And these cells are actually really quite unique. This is an example of one made out of pipe cleaners. This is called a cone photoreceptor, and it's done here in blue because it's going to absorb the sort of short wavelength end of the visible light spectrum, which typically sort of correlates with our perception of blue light. It has, you'll note, like this kind of cone-shaped component at the back. This is called the outer segment. Here's the cell body over here, and this is going to be the axon terminals over here. So it's a very specialized and unique and unusual-looking neuron, which is what, sensory or afferent neurons are. They're always, specialized in some structural form so that they can carry out their very specific, functional roles. And back here, there's a series of membranes, of what they call membranous discs. And embedded in these discs are proteins. These are called photopigments. And they can absorb, different wavelengths of reflected electromagnetic radiation that's arriving here. We're going to see that, there are 3 distinct types of cone cell. Each one expresses a different gene for a different photopigment, which gets embedded in these membranous discs in this outer segment, towards the back of the eye like this. And it's going to absorb it. It's going to change its shape. It's going to then release a chemical. It's going to initiate a series of changes inside this cone cell that will actually, weirdly enough, get it to give a neuronal response, but the response of the photoreceptors to light absorption is actually to stop releasing neurotransmitter. It's weird. They call it the paradoxical dark current, because in the dark is actually when your photoreceptors are the most active. They're releasing the most neurotransmitter. It turns out what they do, though, is they inhibit. It's inhibitory. It prevents output from the eye. So we're going to see, we're going to have 3 of these distinct types of cone cells typically. And then there's another type of cell that's got a big rod-like extension, another type of photoreceptive cell that has many, many, many more membranous discs. And it expresses a particular protein pigment that absorbs basically any electromagnetic radiation within the range of visible light. We're going to see that it is actually really important for kind of dusk viewing or dawn viewing or low light condition sort of viewing. And these photoreceptive cells, the photoreceptors, are located at the back of the retina. So there's several layers of cells. So the photoreceptors are located all the way at the back here with the disc, the cone portion or the rod portion shoved this direction, right? And then this synapse, there's going to be a synapse here, neurotransmitter released onto another cell called a bipolar cell. And they call it a bipolar cell because it's got a cell body. Imagine I'm the cell body, and here's one pole. This is sticking out and receiving the synaptic input, from the cone cell or the rod cell, the photoreceptor. And the other, the pole or projection off that cell body of the bipolar cell is making a synaptic connection with what's called the ganglion cell. And the ganglion cells are located towards the most anterior portion, the front part of this retinal structure here. Photoreceptors at the back, bipolar cell, and then ganglion cell. And then the axons of the ganglion cell will coalesce and form a cable that'll leave the eye at what's called the optic disc. And this is where the axons of the ganglion cells that are carrying information about what the photoreceptors received and then transmitted forward through the bipolar cells. This is going to form what we call the optic nerve, which is going to work its way back, carrying visual information out of each eye. You can also see there's lots of vasculature, lots of blood vessels coming in and out at this particular point. This optic disc is also known as the blind spot because there are no photoreceptors where the axons of the ganglion cells are leaving, carrying information back, and where the blood vessels are, the arteries are coming in, the veins are going out. So the photoreceptor layer is at the back, right? That's where the absorption of electromagnetic radiation occurs. Then the bipolar cells transmit that to the ganglion cells towards the front, and their axons coalesce and form output, via this optic nerve. Now, there are other cells that are located in the retina as well that add complexity to the processing of information as you go from photoreceptive absorption, through the bipolar cells to the ganglion cells and then out. And there are what are called the horizontal cells, which look kind of like octopi with lots of, like sort of projections around, engaging in synapses around a whole series of photoreceptors and bipolar cells. And they add some complexity to the processing of what's going in the retina. And the same is true of what are called the amacrine cells, which are at the second pole, the sending pole of the bipolar cell, where it is releasing neurotransmitter onto the dendrites, of the ganglion cells that'll form the output. So the retina is a complicated structure, and there's some, actual, transformation of the information that's arriving initially at the photoreceptive layer, in these outer segments, of the cone cells, and we'll see the rod cells, that are sort of poked in the eye towards the back this way. Before that information actually, gets sent out the optic nerves and transmitted back to other portions of the central nervous system.